Cholera toxin, also known as choleragen, is a protein toxin produced by the bacterium Vibrio cholerae. It is the main virulence factor responsible for the symptoms of cholera, a severe diarrheal disease. Understanding the comprehensive mechanism of action of cholera toxin is crucial for developing effective treatments and preventive measures. Structure: cholera toxin is composed of two major subunits, the A subunit, CTA, and the B subunit, CTB. The A subunit is the enzymatically active portion, while the B subunit is responsible for binding to host cells. The CTA subunit consists of two functional domains, the enzymatic A1 domain and the membrane translocation A2 domain. The CTB subunit forms a pentameric ring that binds to host cell receptors. Mechanism of action: cholera toxin enters the host cells through a process called endocytosis. The CTB subunit binds to specific receptors on the surface of intestinal epithelial cells, facilitating the internalization of the toxin into the cell. Once inside, the toxin is trafficked to the endoplasmic reticulum In the the disulfide bond between the A1 and A2 domains of the CTA subunit is reduced, allowing the A1 domain to unfold and translocate across the membrane into the cytosol. The A1 domain then activates the G protein alpha subunit, Cs, by ADP ribosylation, leading to its constitutive activation. Activated Cs stimulates the production of cyclic adenosine monophosphate, CAMP, by adenylate cyclase, resulting in increased intracellular CAMP levels. Elevated CAMP levels disrupt the normal balance of ion transport in the intestinal cells, leading to the secretion of chloride ions into the intestinal lumen. This secretion of chloride ions is accompanied by the passive movement of sodium ions and water, resulting in profuse watery diarrhea, a characteristic symptom of cholera. Furthermore, the increased CAMP levels also affect other cellular processes, such as the regulation of ion channels and the activation of protein kinases, which contribute to the overall pathogenesis of cholera. Clinical implications: cholera toxin is responsible for the severe dehydration and electrolyte imbalance seen in cholera patients. The rapid loss of fluids and electrolytes through diarrhea can lead to life-threatening dehydration, requiring immediate medical intervention. Oral rehydration therapy, which involves the administration of a solution containing water, salts, and sugars, is the primary treatment for cholera. Prevention: preventing cholera primarily involves ensuring access to clean water and proper sanitation. Vaccines against cholera are also available, including oral vaccines that provide immunity against cholera toxin. These vaccines can be effective in reducing the severity and duration of cholera symptoms. In conclusion, cholera toxin, choleragen, is a protein toxin produced by Vibrio cholerae that plays a central role in the pathogenesis of cholera. By disrupting the normal balance of ion transport in the intestines, cholera toxin causes profuse watery diarrhea and severe dehydration. Understanding its comprehensive mechanism of action is essential for developing effective treatments and preventive measures against cholera.